Speaker is Dr. Larry Barber. He's going to cover the effects of pharmaceuticals in the aquatic environment. Dr. Barber is a research scientist with the U.S. Geological Survey in Boulder, Colorado, with over 30 years of experience investigating the occurrence, fate, and biological effects of anthropogenic contaminants, including pharmaceutical compounds in natural waters. Please welcome Dr. Barber.
All right, thank you very much. It's really a pleasure to be here today on a snowy Saturday morning talking about one of my favorite topics, which is drugs. And it's really nice to be with an audience of pharmacists because drugs have to be one of your favorite topics too. So I think we're all on the same page. Again, technology is not my friend either. This is that disclosure statement. I don't have any relevant financial interest or relationships pertaining to the content of this program, but I do have a whole lot of interest in sharing the results of my research with you because I'm a federal scientist. I work for the U.S. Geological Survey, and the U.S. Geological Survey is a bureau within the Department of Interior, and our real function is to serve the people of the United States. So when a lot of the questions come up about who is measuring these compounds in the environment, it'll often be the U.S. Geological Survey because we're a non-regulatory agency. We don't have any land management. We are a basic science organization. We provide science. The approach that I take is I'm a geologist by training, so I use geology and hydrology and chemistry and biology, try to integrate them all into interdisciplinary studies so that we can kind of look at these landscape scale processes, and that lets us look at ecosystem health and human health. But really, one of our key factors is that we try to provide science to policymakers and resource managers. Because the way to come to the answer to our solutions, as said by the state regulators, is the more we know about a topic, the better we can manage it. So wastewater is not just some icky thing we have to worry about. It actually is one of the most important future water resources that we have, because with growing population and climate change and demographic shifts, we're increasing our reliance on treated sewage effluents as a major water supply. And one of the biggest issues and challenges associated with this reuse of reclaimed water is the presence of these biologically active chemicals in those effluents. And that really is a major challenge because it's not just a single compound. Today we're going to be talking about pharmaceuticals and drugs, but there's industrial chemicals, agricultural chemicals, and then there's just a lot of biochemistry that comes from people. We produce a lot of chemicals ourselves. So I'm going to start this talk with the premise that, okay, am I not being loud enough? Should I speak louder, but I don't have to speak quieter? Anyway, I'm going to start with this. Okay, something did change my feedback. Okay, like I said, technology is not my application.
Okay, I guess it was better before if you.
So I think you turned off.
I know.
Okay.
The two mics on. I didn't.
So there's.
Okay. So take and then take the picture.
So it's still here.
I know. Let's take it off. If you'll stay close to this mic.
Okay. Well, I can't. Excuse me.
Okay.
Darn. I think I need some. What kind of drug would you use for that? They call it antacids or something. I'm not really sure. Well, anyway, I'm going to start this discussion about drugs in the environment from the premise that pharmacologists know what they're doing, and they design very good drugs that will target some sort of mode of action. So there are a few words I want you to be thinking about. Mode of action. So pharmacology targets modes of action, and what I mean by a mode of action is some sort of evolutionarily conservative cellular and biochemical function. So some of the most effective drugs are the ones that really cover biology. They go after these fundamental modes of action. So the pharmacists and the doctors will identify some need to administer a human therapeutic dose. You give it, however you administer it. And this is how the pharmaceuticals start making their way into the environment. After it's dosed to a patient, then it undergoes metabolism and then it's excreted, which is how it ultimately will make its way into the water supplies. Once that pharmaceutical or drug gets into a water supply, it undergoes a whole lot of different aquatic fate and effect type of phenomena. We don't have time to go into much of that today. But as Barber said in her talk, one thing that's pretty unique about drugs is that they're often designed for recalcitrance. They're designed to be metabolically stable, because if you take the medication and you metabolize it before it has its effectiveness, then it's not a very good medication. So they're designed to be metabolically and chemically stable so that they're pharmacologically effective. Now, one of the things about this mode of action is we've got to be thinking, because it's biologically conservative, it's not just the target organism, which is the human, that gets dosed, but we have to think about these non-target organisms and ask the question, is the therapeutic response a positive or maybe an adverse outcome? So one way we do that is by starting to look at the plasma level, at the body burden of organisms with these pharmaceuticals, and then that lets us start looking at the ecosystem and human health impacts. Well, to be thinking about a mode of action, one of my favorite areas of research is in the human and other vertebrate endocrine systems. Now, the human endocrine system is very important. And again, this deep biological conservation, all vertebrates have effectively the same endocrine system. We all have a hypothalamus, we all have kidneys, we all have thyroids and so forth. The ones I'm going to be talking about today is this hypothalamus pituitary gonadal axis. So if you kind of go from the brain to the pituitary down to the gonads. And these are, what I mean by the endocrine system is it's a whole series of glands that provide a lot of key functions that govern every aspect of a vertebrate. But it's not just the glands. The glands secrete chemical messengers called hormones, and then those hormones interact with some sort of receptor system in the responding organism. So with that as a mind, let's think about how fish might be interacting with this endocrine system. And we'll go back to the hypothalamus pituitary gonadal axis. This is sort of like when I give this lecture to younger people, it's sort of, not that we aren't young, but, to the college crowd, to the undergraduates, you go, well, this is a thing that when your eye sees someone that looks really nice to you, your heart kind of flutters, and the next thing you know, you fall in love. Well, this is the brain is generating a series of biological cascades. The brain, the hypothalamus, starts producing the gonadotropin-releasing hormone, which cascades down and starts releasing gonadotropins, which then start developing the ovaries and the testes, which produce the basic biogenic sex hormones, estrogens, androgens, and the progestins. Now, this gonadal hypothalamus pituitary gonadal axis is really, really important because it governs and controls sexual differentiation, sexual maturation, and the fundamental process of reproduction. Now, this whole axis has been around since humans and basically since vertebrates have been on this planet, and it's driven by some primary regulators. So a fish, its reproductive physiology is driven by the temperature and the length of the day and the surrounding water chemistry, and it's also very importantly driven by social interaction with the other organisms. If you get a fish by itself, it doesn't have any desire to reproduce. Put it in an arena with some companions, and it might change its behavior. But these natural primary regulators, when we talk about something called endocrine disruption, really what we mean, there's a series of anthropogenic endogenous chemicals, such as pharmaceuticals, that disrupt this feedback mechanism that causes these cascades. And what I mean is an example of endocrine disruption, if you take a male fish from certain type of species and expose them to estrogen or estrogen-like chemical, they will start in their liver producing an egg yolk protein called vitellogenin. Now, males normally do not produce this egg yolk protein because they don't produce eggs. But there's that fundamental mechanism that they can do it. So when they're exposed to estrogens, they'll start doing this. And this vitellogenin, I'll get back to it later in the talk, turns out to be one of our really important biochemical markers. So as Barber was saying earlier, when we start talking about some of these more complex mode of action type of processes, we have to think about toxicology in more than one way. We can think about the traditional acute toxicology where you have very high doses and the end member is death or disease. And the nice thing about that toxicology is you normally have a fairly well-defined dose-response relationship. As you get more dose, you get greater effects. Now, when we start looking at the chronic toxicity of things like like endocrine disruption, it's a whole different story because, first of all, the concentrations where you start invoking these biochemical processes is very, very low relative to what you kill things. So suddenly, this part per trillion level isn't just something that us analytical chemists can do because we can do it. This is the level at which chemicals are eliciting biological responses. So we've got to be down. We're not just wanting to go to the part per trillion level because we can. We have to be at that part per trillion level if we want to understand the effects. So again, the other part about this chronic part of this toxicological program is we can't just look at the number of fish that float belly up and die. We have to start looking at very detailed sort of biochemical, physiological response mechanisms. And then the biggest challenge is that with endocrine disruption, these chronic effects, we do not have well-defined safety thresholds, i.e. we often will get a U-shaped response curve. You have a very high effect at low dose. As you start saturating the receptors and sites, so forth, you might have no effect, and then when you get it, so you can have multiple modes of action depending on what the dose is. So it's a very complex situation. So here's my first question for you. It's kind of not an after fact, but it's a more for the fact. What are your favorite pharmacological modes of action? Now, if you haven't really thought about that, I'm going to try to make this easier for you. So there's only three. Estrogen receptor binder. Anyone think that's their favorite mode of action? How about selective serotonin uptake? All right, there we have some. Yeah, you guys are all depressed. Now, let's think about folate biosynthesis. Anyone care about that one? Okay, well, so it suggests that maybe this group has not been thinking a lot about mode of actions, even though your entire profession is built around mode of actions, because pharmacology is a mode of action driven phenomena. Well, oh wow, 83%. Oh, no one likes folate sorry about that one. Well, my favorite mode of action is estrogen receptor alpha and beta binding. Because what's there not to like about sex? Sex is a very interesting topic. It's in the media. You can watch it on TV. But more importantly, it's what drives the world. That if we don't have sexual reproduction, there will not be life on this planet. So one reason I really am fascinated with the estrogen receptor is, there's a lot of reasons, but one is it has very broad cellular distribution. We think estrogen receptor and sex hormones and all of that. Well, you start thinking about the gonads. Well, the estrogen receptor is distributed across all kinds of tissue. And the estrogen receptor actually controls many, many different biological outcomes, not just reproductive. So, for instance, obesity, cardioprotection. But today I'm going to be talking about gonadal dysgenesis and neurodegeneration or neuronal remodeling. Again, these relate back to those mode of action questions. So when I say I'm interested in the estrogen receptor alpha-beta mode of reaction, what does that mean? Well, as I was describing the endocrine distress system, we have these hormones and we have the receptor. So the receptor is embedded in the tissue, waiting until some chemical messenger comes and activates it. Well, the number one activating ligand for the estrogen receptor is, guess what? It's estrogen, which is also called 17-beta estradiol. 17-beta estradiol, the primary female sex hormone. And I'll digress a little bit and talk about the structure. And this is a really important chemical structure. It's been on the planet for about 4.5 billion years. Basically, estrogen is based on the estrane skeleton. It's a fused four-member ring. In this A ring right here, can you see my pointer? Okay, good. Whoops. Here we go. In this A ring, it's a phenolic functionality, and it's got a 3-hydroxy. Now, it's called 17-beta estradiol, meaning that it has 2 alcohols on it. And so you have a 3-hydroxy in this A-ring phenol, and then you have this other 17-hydroxy in the 17 position. Now, when we talk about something like 17-beta estradiol, the beta means its stereochemical configuration. And so this hydroxy right here is sticking up in the same plane as this adjacent methyl unit. And this is actually very important when we start talking about biological activity, because these are very precise interactions. They're not crude, brute force things. It's very specific mechanisms. So whether that hydroxy sticks up or down relative to the plane has a lot of effect on its activity. So this is sort of that natural biogenic female sex hormone that all of us produce, both male and female. But since you're a pharmacologist, one of my favorite pharmacological estrogens is this chemical called 17-alpha ethinyl estradiol. Are you all familiar with that one? Oh, come on, serious, you gotta be. Because 17-alpha ethinyl estradiol is a pharmacological equivalent of 17-beta estradiol. It's the exact same molecule, except back in the 40s they learned that if you go ahead and functionalize at 17 position in the alpha down from the plane with the ethinyl functionality, it makes that molecule resistant to first-pass hepatic metabolism in mammalian systems. And so suddenly you take an estrogen and you make it an extremely effective oral contraceptive. If you don't have that ethinyl functionality there that prevents metabolism, you would have to take birth control pills about that big because your body would very quickly metabolize the active ingredient. So by changing one tiny part of that molecule, we've developed an extraordinarily potent molecule. When I say it's important and a potent, it's not an impotent, but it's an important molecule. Actually, it might be an impotent molecule because the premise I'm trying to make is in the United States, there's about 100 kilograms produced a year of 17-alpha ethinyl estradiol. And yet that 100 kilograms, more or less the size of a human body, completely shuts down the annual reproduction of almost 13 million women. So that is an extraordinarily potent chemical. Now, this is one of our biggest issues we have as environmental scientists, is to get these numbers is almost impossible. The industry tells us that 88 kilograms were produced in the United States in 2007. I go hogwash because in 1998, that was the exact same quantity that was produced in the United States. Our population has grown, the dosing has grown. There's no way those numbers are the same. So how this 88 kilograms gets stuck in the information, I do not know. If we go out and we start taking the amount that's prescribed to the people, the women, just using some crude assumptions, we go, well, you got to have at least 120 kilograms per year. And I know that there are lots of other off sources that no one really thinks about. So these numbers are a real challenge to us because what we try to get after from the regulatory and the fate effect is this maximum environmental concentration. Well, 17-alpha ethinyl estradiol is not the only estrogen that's pharmacological. You know, here's another one, the conjugated equine estrogens. You guys all have to prescribe these because they're very, very high prescription uses. And basically, conjugated equine estrogens, they include all of these traditional things like 17-beta estradiol, 17-alpha, but they also have these B-ring unsaturated horse estrogens or equine estrogens. Now, this is a complicated formula, and it's basically because it's prescribed for postmenopausal hormone replacement therapy. There's a larger patient base, and the doses are much higher than for ovulation inhibiting. And so the annual production and use of this pharmaceutical is about 1,700 kilograms per year instead of about 100 kilograms per year. But the thing about it, these equine and other classical estrogens are equipotent. So suddenly we have all these highly potent, biologically active sex hormones floating around in the environment. A lot of them are produced by the people, but a lot of them are actually prescribed to the people. So that gets us right back to this concept of mode of action and what Barber talked about in terms of this acute and chronic toxicity relationship. Well, because drugs are designed for low toxicity and high potency, because you definitely don't want to kill people when you give them medications. And you don't want to overload the system. So they're by design quite unique compared to the traditional health department kind of contaminants. Love Canal, well, you know, that was PCBs. These were industrial chemicals that had inadvertent biological side effects. Pharmaceuticals are exquisitely designed to have magnificent biological effects. So we've got to be thinking about these chemicals quite differently. So let's go back to this 17-alpha ethinyl estradiol. I like to call it EE2. My tongue gets tired after saying it too many times. But again, has a very low acute toxicity, about, I think the number is about 2 milligrams per liter. So that's a pretty high concentration before you'll kill a fish. But it affects the reproduction of a fish at 0.4 parts per trillion, nanograms per liter. Now, this, Barber was going, well, these are down at a part per trillion. The effect level for EE2 is below a part per trillion. So we can only measure a half a part per trillion. So our best analytical methods are barely getting us down to this lower effect level. So again, if you just take that simple acute chronic toxicity ratio, this tells you that EE2 is interacting by a mode of action because that ratio is 4.6 million. That means you have a molecule that is not very toxic, but it has an extraordinary effect, and that effect is because it's interacting with one of these specific modes of action. Now, if you take invertebrates, which do not have estrogen receptor-mediated reproductive physiology, that acute chronic ratio is from 10 to a few hundred. So again, EE2 doesn't affect their reproduction because they don't have estrogen receptors as part of that. Now, we have to also think about, in terms of what the regulatory community deals with, something like a hazards quotient. Okay, there's so much of it out there. How is it good for us or bad for us, or do we have a risk? Well, the hazards quotient is basically if we go out and measure the environmental concentrations and we divide those by this predicted no observable effect concentration of 0.035 nanograms per liter, what we observe is that if you take most of the streams in the country, under their low flow conditions, they have hazard quotients greater than 1, meaning there is a potential risk for reproductive impairment due to these pharmacological type of compounds. Now, this is just kind of a concentration-based effect. So now, if we go back to this mode of action and talking to the pharmacologist again, there's a way we can really start understanding this in a meaningful, predictive fashion. And this is what we call the effects ratio, because in the drug regulation and registration, there's a tremendous amount of, compared to almost all the chemicals we deal with, pharmaceuticals have the most biological information available. It's part of the registration, the toxicity and all of that. So there's a very important parameter that we use from the human therapeutic, it's called the human therapeutic plasma concentration. Again, this is part of the typical dosing regime, and basically if you have a therapeutic plasma concentration, that means that's the level at which you have the effect that you're trying to achieve with this drug. Well, the counterpart of that is we need to be able to go out in the environment and determine the fish or the organism's steady state plasma concentration. And then we can start comparing those two and go, okay, well, is a part per trillion of water going to cause effect in the fish? And that is where, to get this fish steady state plasma concentration, we have to look at the partitioning and the environmental concentration. And this is where a lot of the drugs may be at very low level, but when you take bioconcentration into account and bioaccumulation, their plasma levels might be fairly significant. And this is all important because, again, we're thinking about non-target organisms. We know that human therapeutic dose is good for humans, but we really don't know what's going on with these non-target organisms that it may be affecting. And that's what we call this adverse outcome pathway. And again, it's the adverse outcome pathway due to the conservation of these enzymes and receptors from mammalian to other types of vertebrate systems. Well, there's a lot of assumptions in this type of approach that leaves us quite confused oftentimes. So this will be my next question to you. Do you think that drugs that are less than a part per trillion or part per billion in water do have implications for aquatic organisms? Yes? No? How about some maybes? Okay, well, that's where, because this is all the answers I think that we have are yes, no, and maybe. So it's a very complex situation. We can't look at one given point. Now, the reason I bring up this 1 part per billion level, though, is basically from the FDA regulation of pharmaceuticals in the environment. If you take all the pharmaceutical produced in the country and you take all the wastewater produced in the country and you mix them up in a big swimming pool, if that level is less than 1 part per billion, there is no additional environmental regulatory criteria that have to be addressed in terms of ecological risk. Now, I just talked to you about something like EE2, that its lowest effect level is 4 orders of magnitude below that. And again, this is based on chronic toxicity. Drugs are not acutely toxic, but they have these very important chronic effects. All right, so now we've got that. So let's take this home to some real examples. My lab's up in Boulder, so we've been doing a lot of study over the last decade at Boulder Creek. And it's not just because it's in our backyard. Boulder Creek really offers a beautiful landscape-based experimental system. And that's because right upstream from our wastewater treatment plant, we have a relatively clean, unimpacted stream, i.e. there are no wastewater treatment plants of any size discharging into it. So it's pretty much, quote, pristine or a low-impacted mountain runoff. And then we have the City of Boulder's wastewater treatment plant, which discharges about 50 to 90% of the volume of the downstream, and we go from a clean, pristine to a wastewater-dominated stream over about 100 meters. It's really nice. There's a low-head dam right at that point that prevents fish from migrating up and down. And then we have the common white sucker, which is a species that lives both upstream and downstream. Now, when we were talking about concentrations, again, if we measure 17-beta estradiol in Boulder Creek, at the upstream site, we can't detect it, and at this time, our detection limit was about 0.8 parts per trillion. But in the effluent, it occurs at about 2 or 3 parts, 2.9, or roughly 3 parts per trillion. And then down in the stream, the concentrations of estradiol, 17-beta estradiol, is around 1 or so parts per trillion. You go, well, that's pretty good. It's way below a part per billion, almost 1,000 times lower. There shouldn't really be a problem. But if we go out and sample the fish in that reach of the stream, and this is what we did, a paper we published back in 2008, what we find is that in the fish downstream from that wastewater treatment plant, we have severe reproductive impact, i.e. the female sex ratio goes from, in the upstream site, the reference sites, it's about 1 to 1. Downstream, it gets to be about 5 to 1, 5 times the female to male. About 20% of the fish population, the white sucker, had what we call gonadal intersex, i.e. in one gonad they have completely fully developed ovaries and completely and fully developed testes. This is a sign of chemical signaling causing these organisms to get mixed up during their sexual differentiation. All the male fish that we sampled downstream had roughly a 1,000,000-fold increase in that plasma vitellogenin, this egg yolk precursor, in their blood. These are male fish. They shouldn't be having that. That tells us that they've been exposed to estrogen. And then from the reproductive level, if you look at the sperm, in the reference, all of the fish were highly reproductive. Their sperm was really hot, ready to make little baby fish. But everything downstream, their sperm were non-viable. Again, these fish had undergone significant feminization or demasculinization. Now again, this is an estrogen receptor mode of action adverse outcome because it's adversely affecting the in-stream fish population. Now, the question when you study things in the stream is, and this is particularly important to the state regulators and the federal, is, oh man, there's so many variables. Where'd the fish come? Blah, blah, blah. And we can't answer a question with any sort of definitiveness that we need to have to do something like put in regulations or put in any criteria. So we conducted a series of additional experiments using different fish species, and we did them in a controlled situation. We set up a laboratory on the site. We pumped water directly from the stream and directly from the effluent channel. We used mature male fathead minnows. We did a 28-day continuous exposure. We controlled food, diet, light, all the things that affect reproduction so that the reference in the effluent controlled fish didn't have any environmental variables. It was simply the chemistry of the water they lived in, and we reproduced the same effect. Very, very rapid demasculinization. This is just vitellogenin, but here again, this is our reference in red, and then the blue and the green. You can see about a 5-fold induction, a 5-order of magnitude increase in that vitellogenin in just 7 days. Within 7 days, the male fish, like this is a typical male fathead minnow. It's got this great big bulge on its head. It's called a fat pad. Within 7 days, that fat pad recessed. The males went from butting heads, being very aggressive, to they all congregated in the corner. All classic examples of feminization. And it was extremely rapid. And we attributed it to the chemistry, the estrogens. All of this is mode of action driven. So now we've said, we've got the chemicals, we know there's an effect, what can you do about it? Most people really hate it when I give talks because they go, oh man, I'm so depressed, the world is coming to an end. But again, science is about information. We don't have any problems we can't fix. We can only we only have unfixable problems that we're ignorant of. So what can we do? Now, we're going to be talking about what can we do, and here's an example. The city of Boulder in 2007, so this highly demasculinized environment was from 2002 to 2006. 2007, the city of Boulder spent $50 million and they upgraded the sewage treatment plant from a trickling filter to an activated sludge. Right there. Well, we had fortunately been running these experiments beforehand. We ran the experiments afterwards. And what we found is that $50 million investment completely eliminated or nearly eliminated all of the estrogenic chemicals that were not being removed by trickling filter were suddenly removed by activated sludge. But most importantly, this endocrine disrupting effect in the organisms was eliminated as well. So if you can remove the chemistry, you can remove the biological effects. And in fact, now, 5 years later, after this, the entire stream ecosystem has responded. We don't see any intersex. The male fish do not have vitellogenin. The sex ratio is back at 1-to-1. And more importantly, we're seeing a lot of aquatic species in that reach of the stream that were really never there before. And it wasn't because of temperature or DO or ammonium. It was really because of all of these other, quote, toxic drugs that were affecting them. So, here's another set of questions for you. The estrogen receptor mode of action. It's important because, A, 17-beta estradiol controls reproduction. Come on, that's an easy one. All in favor? Okay. Estrogen receptors are located in a variety of tissues. Yes. Okay, good. Many disease outcomes are estrogen receptor mediated. Would you all agree with that, kind of? And then here's a trick question. I'll give you it away. Estrogenic effects of drugs only impact human targets. No, I mean, if it's going to impact a human, it's going to impact other vertebrates because we have this biological conservation of mode of action. Okay, well, this is really amazing. High tech. So now I want to go into the next mode of action that all the pharmacists here seem to really like, and that is the neuroendocrine system. That is the brain and the selective serotonin reuptake inhibitors. Anybody know what those are? Oh, yes. Well, we must know because antidepressants are widely used in the U.S., and in 2007, they were 6 of the top 200. The ones I'm going to talk about today are venlafaxine and sertraline. There are some of these other compounds like fluoxetine that aren't even on the top 200, and that's because they're off patent. So again, we have, as environmental scientists, we have a tremendous difficulty. And one thing I'd love to come out of this kind of interaction is this source term is very ambiguous, and it'd be wonderful to clean that up. Now again, these topics often get joked. You know, that last summer, the New Yorker. Well, they say it's from all the antidepressants in the water, and that's a happy fish being eaten. I see the same thing where it'll be a fish with, gonads, and they'll go, well, we can't do it, but we don't care. Because really, what we have is where the fish are being exposed to estrogens and having their reproductive physiology disrupted, they're also being exposed to antidepressants and having their neuroendocrine system messed up. So, and for instance, if you take fluoxetine and you expose crustaceans and bivalves to them at about 30 parts per trillion, extraordinarily low levels compared to dosing, they spontaneously release all their sperm. Now, these are bivalve. They get so relaxed, they just let it go. They kind of have an epiphany there, but that's not really good because that's not the way the reproductive cycle is supposed to function. They are not supposed to spontaneously release their spawn. Now, if we go back to Boulder Creek and think about a different class of chemicals than estrogens, we're going to talk about the SSRIs or the antidepressants, and let's start thinking about the neuroendocrine. I said there's a hypothalamus, pituitary, gonadal axis. Well, again, it all starts with hypothalamus, and this is this neuronal reprogramming. That's what antidepressants do. They affect the ability to have a neuronal exchange. And so if we take those same fish downstream, now this is kind of a complicated diagram because this is a normalized axis here, and these are just listing some of the antidepressants, venlafaxine and sertraline. But what we can see is that the venlafaxine is a very water-soluble molecule, and if we go out in Boulder Creek and we take the white sucker and we measure the water in the sediment, we find that all the venlafaxine stays in the water, and we can't even measure it in the white sucker brain tissue. But if we go and look at another chemical like sertraline, we find that we can barely measure it in the water. Its concentration is only about 3 parts per trillion, but it is highly concentrated in the fish brains. And it's highly concentrated in the fish brain relative to the sediment. And this is not a bit surprising because these chemicals were designed to interact and to deposit in brain neuronal tissue. And lo and behold, this is what we see. What we see is about a 500-fold increase in the brain versus water for sertraline. So this is the kind of evidence that we go, well, yeah, these chemicals, just because it's very low in the water, 3 nanograms per liter, 3 parts per trillion, doesn't mean that it's going to be low in the brain tissue where it actually has an effect. Now, antidepressants, we talk a lot about Boulder Creek because I do studies there. It's a beautiful experimental site. But this isn't a local issue. It's a national and a global issue. You can go to states like Minnesota where we looked at, did a statewide study of about 25 different wastewater treatment plants of all different types. And again, if we go back here to the venlafaxine, 96% of the effluents had it at concentrations up to 2 or 3 micrograms per liter. And then 92% of all of the downstream sites also had sertraline at levels that were much lower than the effluent. And this isn't because it was being eliminated, it's just being diluted. You have the effluent going into a stream, and dilution drops the concentrations down. So now that we know these antidepressants do make their way into fish brains, and they are widely present, the question always comes up of, so what? Is there an effect? Do antidepressants, so here's a question we asked with this experiment. Do antidepressants influence predator avoidance behavior? Now, predator avoidance behavior, this is getting to be very fundamental because these are reflective autonomic type of phenomena. If you don't avoid predators, you die. It isn't like, if you don't avoid a predator, you get sick. You do not survive. So this is getting into that core neuronal brain chemistry reflective reaction. You do not think about what you're doing, you react. So what we did is we exposed larval fathead minnows. Again, they either grow up to be big fish or they don't. So it's a good statistical comparison. We exposed them to a variety of different antidepressants, sertraline, venlafaxine, at environmental concentrations that we've measured across the country. And then after 12 days, we took those little larvae out and put them into an arena. Hopefully you can see that. It's a very, very small, tiny fish. And then we exposed it to a stress. Basically, there's an LED light and a vibration. And so we kind of, get that fish here, and it's sort of just hanging out. And suddenly there's some sort of threat. So it's kind of envision you're standing here, and holy cow, something just fell from the sky. Well, that's what that light does. And you can see that this fish basically goes into what we call, this is one of these fast start behaviors. There's something called the latency period of how long does it take from the time that it gets this response until it responds. And then it goes into what's called a C-start behavior. There's they're called Mathian cells that bilateral down the fish, half of the nerves just collapse. And then that fish goes into a C, and then they re-engage and that straightens out. So it's kind of like flicking something off of your finger, or here you have the fish going into a C. As it does its tail, it generates this escape velocity. So if you take the latency period and the velocity, so again, the curvature and the speed with which it straightens back out affects that escape velocity. Well, what we find is exposure of these larvae to different types of antidepressants and mixtures of them affects that total escape velocity. And it decreases predator avoidance. And that's an adverse outcome. Basically, it means those little fish get eaten. Now, let's take this right back to the pharmacological side of things as human therapeutic plasma concentration. And if we were to take fathead minnows, this time we did, Vellante et al. Published this, did mature male fish, exposed them at about 330 and 50, or 310 and 30 micrograms per liter of sertraline. First of all, you can see that as you increase the concentration, the plasma concentration increases, and in every one of these situations, the effects ratio, which is the human therapeutic versus a fish steady state concentration, exceeded 1, meaning even at these really, really low doses, the plasma level in that fish is equal to what would be in the human that was being given that drug. Now, that is what gives us some insight to just because it occurs at low levels in the water doesn't mean you can ignore it. You have to think about how does it partition into the organism. And now again, this is an antidepressant. It's a SSRI. And here's another, Barbara alluded to this earlier, it's another one of these life or death phenomena. These adult fathead minnows, during the daytime, they seek shelter and shade. Envision it, a little minnow sitting out in the middle of a stream. If it sits in the light, something's going to come and eat it, and it won't be there tomorrow. And so during the day, they hide in dark spots, and when the light comes, when it gets dark, they go out and they do their grazing. And at all these doses, sertraline reduces that shelter-seeking behavior. And so the fish that are exposed to these antidepressants, during the light, they hang out in open water. This, again, the loss of anxiety to a fish is likely to result in adverse outcomes. So the final mode of action that no one really seemed to care about or be aware of, and myself included until I started going down this path, is the foliate synthesis inhibition. And that's if we start looking at antibiotics. Now, I've been talking about SSRIs where you take a drug and it affects your brain, or birth control pills where they interact with estrogen receptors and prevent ovulation. Well, with antibiotics, it's an entirely different process. The antibiotics, you're trying to kill an invasive organism that's in your body. And so some of the first antibiotics were the sulfonamides, of which sulfamethoxazole is a case example. And the sulfonamides, sulfamethoxazole in particular, basically they're an analog to para-aminobenzoic acid, and they interfere with this folic vitamin B9, I think is what folic acid is. But they interfere with that particular metabolic pathway, biosynthetic pathway. And so by interfering with that, it's basically toxic. It kills bacteria. But because the folic acid mode of action, biosynthetic pathway, is evolutionarily conservative between plants and bacteria. That means not only is sulfamethoxazole toxic to bacteria, it also has the potential to be toxic to plant, another non-target organism. So just in terms of not only do we have reproductive effects, we have a lot of other kind of implications that go on in terms of subclinical exposures. Now, sulfamethoxazole is usually thought to be a surface water contaminant. Sewage effluents discharge to the streams. But as Barber said, we also have them around septic tanks or on-site landfill, land wastewater disposal. And we've been doing a lot of work up on Cape Cod, and we find that, in fact, we have some groundwater contamination plumes where the sulfamethoxazole concentrations are equal to what we see in modern sewage effluents. Difference is this plume is about 30 years old. It's been in the ground for many, many years. And we started asking some questions because the sulfamethoxazole in that plume co-occurred with nitrate. Now, nitrate is a drinking water contaminant. It causes blue baby syndrome. It interferes with oxygen uptake. And so there's a really important natural process called denitrification where groundwater bacteria will take that nitrate and convert it to dinitrogen gas, basically detoxify one of these anthropogenic contaminants. But if we take those groundwater bacteria and we concentrate them and expose them to sulfamethoxazole, basically what's happening is the sulfamethoxazole selectively goes after the microbial community in these indigenous populations that's performing this very vital ecological function, nitrogen transformation. So again, the non-target organism it targets a bacteria in your gut, but when it gets out in the environment, it also influences these bacteria. Again, as Barbara said very nicely in her talk, once you are modifying once you're toxic to the microorganisms that are doing the work, then you have these secondary effects. So the nitrate levels stay high. So sort of in conclusion, or my final slide here, is it's not just the microorganisms, but if we take that sulfamethoxazole at about 1.9 micrograms per liter, I said that's what we see in effluent, we see it in groundwater, because of that evolutionary conservation of the mode of action, of the interfering with the folic acid degradation pathway, sulfamethoxazole is also very herbicidal, particularly to things like duckweed. And so we can see that even at that 1.9 microgram per liter, that hazard quotient with sulfamethoxazole, if we use a mode of action target, so again, if we use these non-mode of action targets like how many fronds are on the plant or how much does a plant weigh, those aren't very sensitive. But when you get into those biomolecular, physiological mode of action driven effects, you can start seeing dramatic responses in terms of as the para-aminobenzoic acid increases, that basically means the folic acid synthesis pathway is being shut down. So by thinking about how pharmaceuticals are designed to interact with humans, it really gives us, I think, a lot of potential to deal with the problem, because we know a lot about how these chemicals are interacting. We know what's causing their effects. Now we just have to work at how to minimize their exposure in the environment. So that would be this. I don't know really if this is the last question, but it's one that is sort of the take-home message of whether it's a pharmacist or an individual. You know, let's start all thinking about what can we do to go after what Joe was saying. Let's go to the top or whichever pyramid he wants. Let's do the minimization. The problem with drugs, they're often overdosed. So again, I think we can minimize and this is, we did the exact same thing with hazardous waste. A chemical that you guys are talking about now, nonylphenol, a synthetic mimic to these things. You know, if people start using proper dosing, then the loading goes down. If people quit disposing, the best thing that we can do, the most important thing, don't consider the toilet and the sewer as our hazardous waste disposal mechanism. It's not designed to do it, but that also gets into I've heard some of the craziest things being thrown out there as solutions. And so I think there's a lot to, like for instance, when I say crazy, I read once that the DEA said, take all the drugs and mix them with chlorine bleach. Now, has anyone ever heard about, maybe if you've never cleaned, it's like, don't ever mix chlorine bleach with Mr. Clean. Because you mix chlorine and ammonium, you produce toxic gases. So you've got this extremely complex chemistry, and the DEA says, pour chlorine on it because that's going to make it unsuitable for someone wanting to take your opiates. But you go, what on earth are we generating when you start taking highly reactive things and doing it? And then the down part of it, pour chlorine and then put it in your garbage can. Anyway, I think this is an incredible opportunity to have people sitting and thinking about sound solutions. Understand the science and come up with good solutions, not just reacting to issues. And that's a very I think, as you said, Joe, I mean, and Barber as well, this doesn't come quick. I've been working on, this for like 25 years. And finally, I got an audience with pharmacists. Normally, I'm talking to people that don't even know what a pharmaceutical is. So anyway, thank you very much. Any questions? I'm not the only one that gets to ask questions, so come on.
I don't mean that to be insincere or to be silly, but has your work changed your view on what kinds of human reproduction, birth control pills versus other forms of reproduction should be in vogue, for lack of a better word? Should we stop using oral contraception and go towards other means based on the the effects that they have, the E2 and whatnot?
Well, I think there are alternative non-oral contraceptive, again, I'm not an MD or a pharmacologist, but there are alternatives which could help with the problem. Again, I think one of the real challenges that we have in the area of dealing with drugs and pharmaceuticals in the environment is you always have to balance it with the human health and societal benefits. Now, if you have some industrial chemical that's being dumped out there, it's I think as the health department said, it's a no-brainer. This is a problem, we can fix it. But with the pharmaceuticals, we always have to be looking at the cost benefit. And that's where, to me, I think one of the things that I've seen evolving over the last, say, maybe 10 years with the pharma and the pharmacological industry is the idea of going with green design. So, it kind of started out with, well, we can't quit prescribing drugs because people will die or will overpopulate the planet or some other kind of component. It's like, well, but can you design a drug that has the same effectiveness that doesn't have the same environmental persistence? Now, we've done this time and again with industrial chemicals. You take herbicides, or pesticides, insecticides. We used to use DDT. That's still around. It was designed to be absolutely persistent, and it did an extraordinary job, but it didn't go away. Same way with many of our drugs, they're designed for persistence for this metabolic component, but I think Barber hit on one of the things that I think is very important. Maybe we can be designing drugs that are photolytically labile. They're not biologically labile, they're not metabolically labile, but as soon as they get hit with UV light, they undergo a transformation, and you don't have to destroy the molecule, you just have to get rid of its effectiveness. So again, that's what I see is there's a lot of opportunity with creative design, and it's not a one or the other. I don't see there's any, we were able to, the industry was able to design better surfactants, better pesticides. So the pharmaceutical industry is, to me, one of the most brilliant industries on the planet in terms of the ability to manipulate chemistry. So I see a lot of great potential here.
Thank you.
Any more questions? Well, thank you very much.